Namaste friends, welcome you all for the biology session. This is MGS biology lecturer here. Let us continue with the chapter microbes in human welfare. The first part we discussed what and all the roles of microbes in household products and industries and obtaining organic acids, and along with that certain enzymes and bioactive molecules. So now let us move on to the STP, that is sewage treatment plant. So microbes are also play a major role in the treating sewage, as we all know that municipal waste, right? So municipal waste, right, which includes water and also solid waste. Almost all we can say that the both water, that is a liquid, and solid waste, which we can consider as municipal waste. So this is mainly which includes a large amount of domestic waste, that is what we can say human waste, along with the animal excreta and microbes. So this is what we can say that the sewage. So generally to treat that the municipal waste and along with that large amount of domestic waste, right? So domestic waste together we can consider as the sewage. So in which we can say that generally the biological oxygen demand will be very high due to the presence of more organic matter. So in this mainly STPs to treat these this sewage, we're going to use STPs, sewage treatment plants. So sewage treatment plants which are used mainly to treat sewage, to convert that which is there more polluted to less polluted. That is where we can say sewage treatment plants used to convert that is more polluted, right? So more polluted sewage to the less polluted. So that is the major the significance or role of STPs. So that is by using, here we can say that we're going to use by using a microbes which are heterotrophic microbes. We're going to use heterotrophic microbes, right? So in this generally we have two stages, right? In the STPs, we're going to treat the sewage in two stages. One is a primary treatment. So in the first stage, that is which includes a primary the treatment. In this primary treatment we can say that is mainly which is a physical treatment. So primary treatment which is a physical treatment, that is removal of floating debris, right? Where you can say that the sequential filtration will be there. Sequential filtration and sedimentation is a major step in this. That is sequential filtration and sedimentation. First we're going to remove the floating debris in this sequential filtration, and the sedimentation of the solid waste. So sequential filtration and sedimentation which included in the physical the treatment, which is also called as primary treatment. So in this mainly we're going to remove floating debris. After removing floating debris, we're going to remove which will going to sediment. So debris waste will going to settle down. And here the secondary treatment, primary treatment is called as physical treatment, primary treatment is called as physical treatment, and secondary treatment which is called as the biological treatment. So in this biological treatment mainly we can say that where in which we're going to they remove, right? So mainly the waste using the microbes, right? So in this we have to keep it in mind. So physical treatment which includes removal of debris by sequential filtration, and finally the removal of the soil and small pebbles, right? So soil and small pebbles by the process of sedimentation. So let us take, I can say that when in the primary treatment we're going to treat, so first thing is that where almost all the solid is sedimented. So all solids which are sedimented, that settle, right, that forms solid settled, and that forms a primary sludge, and which is the filtration from liquid form, supernatant, right? Supernatant, that is what a liquid, the state which will be there in the liquid state or a liquid form. Supernatant forms, right? Supernatant forms which is we can say that the primary effluent. Now this primary effluent will be subjected to secondary treatment. So there the secondary treatment, which includes what is that, the step by step. So in primary treatment, only physical treatment. So that is primary effluent will be formed and primary sludge, that solid waste which will be settled, that is the primary, what is it, the sludge, and that effluent is the supernatant that forms a primary effluent. So next, in this case, generally we are going to take the primary effluent which will be there, that will be treated with secondary effluent. So primary effluent, secondary treatment, sorry, primary effluent treated with a secondary treatment. What is that? That will be left, that will be, what is it, transferred to the aeration tank. In this aeration tank, mainly we are going to give, what is it, the mechanical support, that is a constant agitation will be there. So in this constant agitation, we are going to supply more oxygen. We are going to supply more oxygen, due to which generally the flocks will going to form. So in one more that is one very important question we can get. In aeration tank, the flocks formation takes place due to the continuous agitation. So what is that? What are those flocks? So flocks are generally the aerobic heterotrophs, aerobic heterotrophic microbes, which are going to grow, right, in the form of, what is it, the masses. So masses and mesh like sheath, or you can say sheath. So that is what you can say, growth of aerobic microbes, right, aerobic microbes, mass of aerobic microbes. So that is what we are going to say, that is the flocks. And in this case, we can expect that one more after, what is it, the forming flocks, which includes that aerobic microbes, and along with that we can say aerobic heterotroph microbes, right, and along with that, which mainly forms a where you can say that the microbes which includes both bacteria and fungi. So that is why in simple way we can take, that is a flocks which are the mesh like structure, right, mesh like masses of bacteria, which is associated with fungal, what is it, the filament. So fungal filament plus mass of bacteria, which forms a flocks. So what is the role of flocks? Flocks which will going to reduce, right, they consume, mainly it will going to consume organic matter, right, that will be reduces, what is it, the reduces biological oxygen demand. So when biological oxygen demand is reduced in the effluent, that is in the primary effluent, so then that will be treated, right, treated in the settling tank. So what happens, that is in the settling tank they're going to remove, right, that is here in this, what is it, this settling tank, they're going to, what is that, mainly I will take step like this. So first you have to keep it in mind, that is a primary treatment, and then to the aeration tank, primary effluent to the aeration tank, and in the aeration tank flocks formation takes place, and from the aeration tank that will be directly to the settling tank, that is the, what is it, the third chamber. And in the settling tank we are going to, what is it, the remove, where you can say that the mainly sedimentation of flocks to form, what is it, activated sludge. So here sedimentation of flocks, right, so sedimentation of flocks which forms activated sludge, we can expect this question. So activated sludge which will be formed in which tank? That is in the settling tank, that is sedimented flocks. After sedimentation of this, so little part of this, what is it, the sedimented activated sludge, which will be used as a inoculum. So this is also very important. So inoculum as a starter, this part, so this part will be used, and that will be, what is it, transported back, right, so where we can say that will be transported back to the aeration tank. So aeration tank, where that in that culture we can increase large number of flocks if we are going to use this inoculum, right. So after that what happens, that is the anaerobic sludge digester, and all these, what is it, the microbes, that is both fungi and bacteria, which will be treated in aerobic sludge digester, so that is what anaerobic sludge digester, which is a large tank, right, in which all those they're going to remove a bacteria, right, that is where we can say they're going to use anaerobic bacteria, and these anaerobic bacteria which are going to digest the bacteria and fungi, digest the flock directly, which in that flock we know that both it includes mass of bacteria and filamentous fungi. That is what we can say, fungi, which will going to digest. So that is why it is called as anaerobic sludge digester. And finally, from that anaerobic sludge digester, during this process, when the digestion takes place, biogas will be produced. So this biogas, which will be with a major constant like methane and hydrogen sulfide, and along with that carbon dioxide, which will be there. So this is what which will be going to obtain. So production of gas is also done in sewage treatment plants. So this is very, very important. So this is what this sewage treatment plant. So next we can say that is the biocontrol agent. They're going to use they generally, in case of biocontrol agents, I've already mentioned that in case of the microbes, where they will be used biocontrol agents, which includes they generally controlling a plant disease and pest. So biocontrol agents means that is biological methods in which they're going to control plant disease, controlling plant disease and plant diseases and pests, which are called as biocontrol agents. Those biological methods or attributes which will be used, natural agents. So here we can mention animals, some plants will be used as a biocontrol agent, bacteria, viruses and fungi, which destroy or control the weeds, insects, pathogen. So biocontrol agent may be biological methods, right? Biological methods which are also include natural agent. So natural agent means some insects, natural agents, right? So insects, and in that they're going to we can say that some insects and plants, animals, bacteria and viruses are also used to obtain which also acts as biocontrol agent. So natural agents includes insects, plants, animals, bacteria and viruses. Along with that, some fungi also used as a the biocontrol agents. So in which they're going to destroy the weeds, right? Some which will be used to destroy, or you can say that they control weeds and also to kill insects, other harmful insects, along with that pathogens. So these are generally the pests. So that is why we will consider as biopesticides. So these natural agents which will be used as biopesticides. So this is what you have to keep it in mind. And best example here in the natural, the control, ladybird beetle. Always we're going to get the question, ladybird beetle, which feeds on the aphids, that is which sucks the plant sap, mainly ectoparasite, and dragonflies which feeds on the mosquitoes, several the larvae, right? The mosquito which acts as a vector. And in this we have studied, that is a BT cotton. I have mentioned that Bacillus thuringiensis. So Bacillus thuringiensis, right? Bacillus thuringiensis, they're going to obtain from this, they're going to obtain the pesticide. That is where we can say microbial pesticide, and these are the microbial pesticides. So the pesticides which are obtained by Bacillus thuringiensis, that is a bacteria, right, to kill the cotton bollworm and other aphids, or you can say other insectivores, arthropods, right? And one more microbial pesticides, that is baculoviruses. I already mentioned solving one question in the previous, baculoviruses, which mainly belongs to nucleopolyhedrovirus. So mainly belongs to genus nucleopolyhedrovirus. So with all together they're going to they used as a IPM. IPM. So and in this we discussed, that is generally the biogas. Also they're going to obtain using the gobar, right? So this is which will be used for heating, cooking, lighting, right? So which is very eco-friendly and pollution free. We'll take that. So here, just in simple way, we can say that the biogas which is obtained using microbes, right, that is we can say that methanogenic bacteria. So to obtain the biogas, they're going to use methanogenic bacteria, right? Mainly we can say this is a archibacteria, right, which includes bacillus. Other bacteria are also there, we can also say that the bacillus which will be used, and what is that, cellulomonas and clostridium will also used. And this for the first time in India in collaboration with IARI and what is it, the Khadi and Village Industrial Commission. Khadi and Village Industries Commission and Indian Agriculture Research Institute, both together they developed this biogas plant. So in this, the one major question we can get, the major constituents of biogas is methane, 50 to 70% methane will be there, and carbon dioxide which will be there 30 to 40% which will be there, and along with that the hydrogen, what is it, which will be there, generally we can say something around 1 to 5%, and remaining which will be there, that is which includes traces of oxygen will be there, and nitrogen which will be there, and hydrogen sulfide also will be there. So this the major constituents is what is it, methane. Right. So and then already we mentioned some microbes which will be used as a biofertilizers. So biofertilizers which are there, these biofertilizers which shows, or what is it, the mainly which includes one, that is the rhizobium which helps to fix the atmospheric nitrogen. So fix, what is it, the atmospheric nitrogen. So which is generally we're going to, what is it, they see in the leguminous plants, that is association between, right, symbiotic association between what is it, the microbes, that is bacteria and higher leguminous root plants which will be there in the pea and beans, like leguminous plants, right. And then the second one, that is Mycorrhizae, we are all familiar with that. The Mycorrhizae, the one which is, what is it, the symbiotic association between fungus, right, fungus and higher root plant, higher root plants, right. So that is, you can say roots of higher plant roots, or you can say higher plant roots, right, where you can say that the root nodule formation can be seen. And mainly the members of these, what is it, the fungal, most of the fungal members which belongs to the genus, what is it, the glomus, it belongs to the genus glomus, right. So glomus is actually the members of, what is it, the fungi, the most of the fungi members which belongs to genus glomus, right. So that is what you have to keep in mind. And which fix the phosphorus, that is very very important, which fix the phosphorus, right. So phosphorus in the soil, which fix the phosphorus in the soil. So this is, you have to keep it in mind. So and the third one which is we discussed, that is a cyanobacteria, right. So cyanobacteria, which is also we can consider as a blue green algae, right. So blue green algae in which autotrophic microbes, that symbiotic association with plants, right. So cyanobacteria which includes Anabina, Nostoc and Acilitoria, right, association with symbiotic association with plants like Anabina, right. So Anabina, Nostoc, and then one more very important, that is Acilitoria. So here we discussed that they helps to fix the, some which helps to fix the atmospheric nitrogen, mainly in the what is it the paddy fields, right. In paddy fields they are going to use, what is it, the regularly they are going to provide with azolla, right. So azolla pinnata which forms a best association with blue green algae. And next, fourth one, that is some are free living, right. In all these above mentioned, that is cyanobacteria, mycorrhizae and rhizobium are the, what is it, association, that is association will be there with the other organism. Here some are free living, what is it, the microbes which fix the atmospheric nitrogen, atmospheric nitrogen. They are mainly includes, what is it, the azotobacter, right. So azotobacter, and along with that which includes azospirillum, right, spirillum, right, and you can say that clostridium. So generally these are the saprophytic bacteria. So this is what you have to keep it in mind. So and finally we can say that is one biofertilizers. If you consider that, the very important thing, the free living nitrogen fixing bacteria which are generally present in the soil, right. So that is very very important. Acetobacter, bacillus, and these are saprotrophic. And where you can say symbiotic association, rhizobium, which forms a, what is it, generally we can say that which forms a nodule roots of leguminous plants. So in this we can say, right, nodule formation will be there. Nodule formation can be seen in this. So and next we can take, that is after this, biofertilizers, right. So in this, the very important thing we have discussed. So let us take some questions related to this. So after, what is it, the question number 15, so we can take, so this is what a biogas plant, right. So here there will be a inlet through which they are going to feed a slurry which includes, what is it, the water and cow dung. So that is inlet. So one inlet will be there and two outlets will be there. So this is the outlet and two outlets will be there in this. So in this question we can expect that the given figure represent typical biogas plant. Select the correct option A, B, C. A is an inlet for cattle dung. So A is an inlet, that is correct. And C is an outlet for the release of biogas. No, this is for sludge, right? Spent slurry, we can say. Spent slurry from which biogas is obtained. And here we're going to, what is it, the generally the methanogenic bacteria will going to help in, what is it, the synthesis of biogas, production of biogas. And this is a gas holder, that is the gas holder, right. So and B is a chamber which contains leftover slurry. No, that is leftover slurry which will be there in this C, right. And then all of these. So here, which respectively you have to select the correct option, right. Where we can say that is the correct answer for this. Here we can take where inlet A is an inlet for cattle dung, right, and outlet, this is for gas, right. So this is where we can keep it in mind. Which of the following option contains the end products formed during anaerobic respiration in yeast? So anaerobic respiration in yeast, generally we can say that the we, in this case, the yeast which will be used to, what is it, they obtain mainly ethyl alcohol, where the ethyl alcohol which will be obtained, their carbon dioxide will be produced. So carbon dioxide, so once we are going to consider that the glucose, right, so where with the help of the fermentation, glucose in the absence of oxygen, which will be converted into generally the water, carbon dioxide, and what is it, the C2H5OH, ethanol will be obtained plus energy will be obtained. So that is what we can say alcoholic fermentation, which is called as, what is that, alcoholic fermentation. But that is by brewer's yeast, right. So this is what you have to keep in mind. So 17th answer is C. Next, wine, beer are produced directly by fermentation, whereas brandy and whiskey require both fermentation and distillation. That is correct, we can say. This is because generally the fermentation is inhibited an alcohol level of 10 to 18%. So when fermentation takes place, alcohol level will be very less. When distillation will be done, that is alcoholic, what is the level, so alcohol level which will be increased more than 10%. So in this, what is it, the whereas brandy and whiskey, that is more than 10%, hence distillation should be done. So here we can say that distillation prolongs storage. No, that is wrong. Distillation improves quality. No, distillation purify the beverage. So if you consider that in the 18th question, where we can say that the fermentation inhibited at an alcohol level, what is it, 10 to 18%. Hence, what is it, we have to go with the fermentation and distillation, right, where beer and wine which will be less than up to, what is it, the less than from 5 to less than 8 which will be there, the percentage of alcohol in wine and beer, because of, right, absence of distillation. The mosses of bacteria held together by slime and fungal filaments which form a mesh-like structure. So that is where we mentioned that in the aeration tank, just now I mentioned, that is in the aeration tank, flock formation will be there. So that flock formation, which is generally represents the masses of, right, so masses of bacteria, right, along with that filamentous fungi. Filamentous fungi. So that forms where you can say that flock. So 19th we can say answer is flock. So primary sludge, that is in the during primary treatment, so which will going to settle down. So that is the solid waste which will going to settle down, sedimented. What is it? The settled solid waste in the primary treatment is called as primary sludge. So answer is flocks. So activated sludge which will be formed in the, what is it, generally aeration tank. So once it will settle, that is in the settling tank, flocks which will going to settle. So settled flock which will be acts as activated sludge, right? So anaerobic in the anaerobic sludge digester, right? After complete digestion, the solid which will be there, that is anaerobic sludge. So answer is B. The process of biogas production is, so already I mentioned that when there will be a biogas production takes place, there is no requirement of oxygen, right? So in this we can say when the process takes place, so these are the methanogenic bacteria, right? So that is methanogenic bacteria, right? And also we can say that the clostridium sometime can be used to obtain the biogas. So the biogas which will be obtained using methanogenic bacteria by the process of methanogenesis, and sometimes the bacillus and cellulomonas also will be going to use cellulomonas. So these which are there anaerobic process, they have a capability to survive in, what is it, anaerobic condition, right? So answer is B. So next, biofertilizers are organisms that enrich the nutrient quality. So I've already mentioned that, like mycorrhiza, rhizobium and free living, what is it, the bacteria, free living nitrogen fixing bacteria like saprotrophic bacteria, azotobacter, bacillus and clostridium, which are the biofertilizers. They enhance the nutrient quality of the soil. Which of the following can be used as a nitrogen fixing cyanobacteria can be used, nitrogen fixing bacteria, that is also used, mycorrhiza can also be used, right? And along with that we can also use, what is it, the free living bacteria. We can also mention that the free living nitrogen fixing bacteria. So nitrogen fixing cyanobacteria means where we are going to mention that is association. Cyanobacteria, that is a blue green algae, symbiotic association with other plants. So algae, blue green algae with, what is it, plants, best example with the rice plants. So that is what Azala pinnata, right? So this is what which helps to fix the atmospheric nitrogen. And nitrogen fixing bacteria, where we can say that is generally in case of mycorrhiza, that helps to fix the phosphorus, and rhizobium which helps to fix the nitrogen, atmospheric nitrogen, right? Mycorrhizae. So 21, the answer is all of these can be used as, so that is what generally we can say symbiotic association, free living will be there, and also association with the plants, association with higher plants, roots of higher plants, and also roots of leguminous plants, right? Roots of leguminous plants. So all these can be used, what is it, in this, what is it, generally. Next, match column 1 and 2 and select the answer for the given codes. So Ganga action plan to reduce the, what is it, the pollution, river pollution. That is Ganga action plan, which is under the Ministry of Environment and Forest, right? So that is, they're going to take care of that Ganga action plan and Yamuna action plan. So they're going to launch this program to take care, to reduce the pollution. So where we can say that the A2, right, and BT cotton, that is a Bacillus thuringiensis cotton, that is insect resistant plants, and B3 can be, you can select B3. So A2, B3. So in this, the first option we have, whereas here it is wrong, and here we have, but B3, B4 we can eliminate it, and A1, B3, that is also we can eliminate. So first option, let us take next options. See, Rhizobium, already we have discussed that nitrogen fixing bacteria. Right, that is not association with cyanobacteria, that is nitrogen fixing bacteria. Cyanobacteria anta ಹೇಳಿದ್ರೆ you have to keep in mind that blue green algae, Anabina, Nostoc, Acilitoria. Right, Anabina, Nostoc, Acilitoria. Right, Azolla, Anabina association. Right, best example, Azolla, Anabina association. Anabina association is the best example for nitrogen fixing bacteria. Right, sorry, Azolla Anabina association is the best example for nitrogen fixing cyanobacteria. Fixing cyanobacteria, that is blue green algae. Sorry, that is with the blue green algae. Whereas nitrogen fixing bacteria, here we can say that the generally best example Rhizobium, nitrogen fixing bacteria, that is Rhizobium. So that is what we can say, I already mentioned here. Then Nostoc, which is there generally nitrogen fixing cyanobacteria. So answer for that is A. Right, so answer is A. Which of the following mismatch with respect to the organism and acid produced by? So already discussed that Clostridium botulicum. So in this case, what is it, the wherein which the Clostridium botulicum, which will be used butyric acid, not gluconic acid. So that is wrong. So answer is, this one is mismatched. Let's see next one. Lactobacillus acidophilus, lactic acid, correct. Aspergillus niger, citric acid, correct. Acetobacter aceti, acetic acid, correct. So answer is, the mismatched one is Clostridium botulicum. Right. That which from which they're going to obtain butyric acid. So butyric acid. Next. So 24th question we can take. Identify the blank in spaces A, B, C, D in following table and select the correct option. So bacterium, right, which is used to obtain streptokinase. So in this 24, right, that is streptococcus, which will be used, streptokinase, that is streptococcus. Coccus anta bantu andre ನೆನಪಿಟ್ಕೊಳ್ಳಿ, bacteria that is used to obtain streptokinase. And when Aspergillus niger, so Aspergillus niger, so Aspergillus, which is generally that is fungi, right, that is fungus, which is obtained, used to obtain citric acid, correct. Fungus Trichoderma Polysporum, that is cyclosporin A. Cyclosporin A. That cyclosporin A, which is a bioactive molecule, which will be used mainly, what is it, as a generally to reduce the cholesterol level, right. So that is where we can say cyclosporin A, that is a bioactive molecule which is used to, what is it, mainly to so that is lowering cholesterol level, which will be used, right. So that is what you can say. Generally in this, the one very important thing we have studied, right, where we can use the pectinase, lipase and streptokinase as a fibrolytic enzyme. Cyclosporin A, that is bioactive molecule which is used, that is to reduce the cholesterol level, sorry, mainly to treat the patient. Statin will be used, so cholesterol, lowering cholesterol level agent, that is statin. So which is obtained using yeast. Statin obtained using yeast, sorry, that is Monoscus purpureus, which acts as the lowering cholesterol agent. Acts as, what is it, lowering cholesterol agent, sorry, lowering cholesterol agent. Whereas this one, cyclosporin A, that is used to, what is it, the in organ transplantation, that is immunosuppressant. What is it, suppressive agent. Immunosuppressive agent. So immunosuppressive agent, which will be used as immunosuppressive agent. So next, bacterium, which is used to obtain the butyric acid. So that is mainly Clostridium botulicum. So Clostridium botulicum. So let us fill it. First, A is streptococcus. In these three options we have. B is fungus. Here we don't have in all those things, so we can select B. That is the A is the correct answer. So cyclosporin A, C, Clostridium botulicum. So 24th answer is A. And next one more, that is Just now we discussed that is a fungi which is used to obtain cyclosporine A, right? Trichoderma, what is it? Polysporum. So 25th we can take, that is Trichoderma Polysporum. Trichoderma Polysporum used to cyclosporine A. So medical application as an immunosuppressive agent. Just now I mentioned that is which can be used as immunosuppressive agent. So this is very very important. And Monascus purpureus, which is used to obtain statin, this one is a yeast. Even this is also fungi but is unicellular, right? So yeast, unicellular, right? So that is why we can say that the C, which is yeast, A is Trichoderma Polysporum, B as immunosuppressive agent, and C is yeast. Statin, which will be used as, just now I mentioned that statin lowering cholesterol agent, lowering blood cholesterol agent. So answer here Trichoderma Polysporum we have in this option, but lowering blood cholesterol level. So hence it is wrong. And yeast, that is wrong, A. And Streptococcus, that is also wrong. So answer for that is A. So A is the correct answer for this 25th question. So given below the flow chart sewage treatment, identify A, B, C, D. So already I have mentioned that primary sludge and effluent, which will be large aeration tank, and where A, what we should do, that is where in the continuous supply of oxygen should be there, right? So oxygen should be there for the development of the flocks. So in this case generally we are going to take, that is the mechanical agitation, that is aeration, mechanical agitation, right? Stirring, and flocks will be formed, and flocks which will be left generally when the flocks formation takes place, that is in this case the BOD will be reduced, and that will be transferred for the sedimentation to the generally to the, what is that, sedimentation. After in the sedimentation settling tank, where here we can say that the activated sludge will be formed. So activated sludge which will be going to form, and after activated sludge that will be treated with anaerobic sludge digester. So anaerobic sludge digester. So in this we can say that anaerobic sludge digester which is used to obtain the biogas form. So that is mechanical agitation, and then reduce BOD, here increase, wrong, reduce BOD, activated sludge is wrong, and B is this one. So mechanical agitation, reduced BOD, activated sludge, anaerobic sludge digester. So answer is B. So for 26th question, answer is B. Next, living organisms used to enrich nutrient quality of the soil are called as. So already I mentioned that which will be used, right? So enrich the nutrient quality of the soil, they are called as biofertilizers. Biocontrol agents, they are the methods which will be used as a biopesticides, right, to control the insects, arthropods and other organisms, right? And then synthetic fertilizers, they are chemical. Natural fertilizers, where we can say that we can use, that is what, Cyanobacteria, right? Symbiotic association. So that is answer for that biofertilizers. It is you can say which enrich nutrient quality of the soil, they are called as biofertilizers. So 27B. So fill in the blanks, this correct option. Biogas is a mixture of gases which predominantly contains, that is methane, and generally which will be used as fuel, that is for lighting purpose they are going to use, and electricity and heat for the to cook food. So that is which will be used as a fuel. So methane fuel, right? 28th question we can say that the fuel. And methanogens are commonly found in anaerobic sludge, that is anaerobic sludge during sewage treatment. And after that the dash or free living fungi, effective in biocontrol agent of several plant pathogen. Methane fuel, anaerobic, what is it, the sludge, free living fungi. So baculoviruses, and that is trichoderma or free living fungi which will be used. So answer methane fuel, anaerobic sludge and trichoderma. So answer is A for this. So 28th question, so answer is A. And 29, Which of the following bacteria is present in rumen kettle? So that is what rumen of kettle, that is a chamber, right? The stomach chamber, right? Rumen, reticulum, omasum and abomasum. There are 4 chambers, right? Out of which generally we know that generally methanogenic bacteria, that is called as methanobacterium. So which is required for methanogenesis, right? So methanogenesis. So 29th answer is C. Right, so acetofactor, free living nitrogen fixing bacteria, rhizobium, association with the leguminous plants, right? Leguminous plants like pea and plants. Azospirillum is also like acetobacter. These are same, free living. So this you have to keep it in mind, that is methanobacterium, which is a bacteria present in the rumen of the cattle, which helps in the production of methane through the process of methanogenesis. A microbial biocontrol agent that can be used to control butterfly caterpillars. So that is what we can say, that the cotton bollworm, they are going to control the cotton, the bollworm, right? And along with that, they also used they control the butterfly caterpillars. That is what microbial control agent, which is obtained from, microbial means which is obtained from bacteria, or you can say that the baculoviruses. So that is Bacillus thuringiensis, right? Trichoderma Polysporum, right? So that is, I already mentioned, that is a bioactive molecule to obtain Streptococcus mycorrhizae. So this is wrong. Bacillus thuringiensis, that is to obtain Bt toxin, right? So that is one, you have to keep it in mind. Next, in this, during the primary treatment of sewage, solid particles that settle down are called as. So during primary treatment, so that is where I mentioned that in the primary treatment where the solid waste that will going to settle, that is called as primary sludge. Primary sludge, and effluent is called as primary effluent, right? So primary sludge, flocks in the aeration tank, activated sludge which is settled, that flocks and settled, that is called as anaerobic sludge in the anaerobic sludge digester. So what is that? The primary during primary treatment, the solid particle which will going to the settle down, that is called as primary sludge. So answer is B. So 32 answer is B. And then study the following statement and select the correct one. Study the following statement and select the correct one. So in this, methanogens are archaebacteria. I already mentioned that yes, they are archaebacteria which can survive even in the harshy condition, right? Which produce methane in marshy area. That is what that harshy condition, that is a marshy area. Study the select correct one. So this is a correct one. And then nostoc filamentous blue-green alga which fixes atmospheric nitrogen. Correct, blue-green alga, or we can say that is cyanobacteria, right? Cyanobacteria. Mycoplasma lacks cell wall and can survive without oxygen. That is also correct. So all the statements are correct. 1, 2, 3, right? PPLO, right? Pleuropneumonia like organism, that is mycoplasma, smallest known living organism. Right, so which of the following organisms is used in production of beverages? So already I have mentioned that the beverages, brewer's yeast, will say, that is also what is it, the same, brewer's yeast and baker's yeast, right? Brewer's yeast and baker's yeast both are same, that is Saccharomyces cerevisiae. So 34th, the answer is B, Saccharomyces cerevisiae. So here this is what we can say, that is Penicillium notatum, from which generally that is they obtain the penicillin, right? So next, 34th, microbes are generally present in soil, thermal vents, RC condition, polluted area, and all of this. So in all of this, that is where we can say these are cosmopolitan. That is cosmopolitan, they present everywhere, right? So that is what you have to keep in mind. Which of the following microbes is proteinaceous infectious agent? So proteinaceous infectious agents, right, which are generally, whenever we're going to say, that is the, I mentioned that viroids and what is that, the viruses and prions. So these are proteinaceous infectious agent, which are prions, right? So prions are activated agents. Prions are activated, what is it, the, we can say, proteinaceous infectious agents, right? So column 1 and 2 and select. So methanogens, generally where they exactly they are present, we can say that is production of methane and fermenters, large vessels, what is growing microbes which will be used to obtain antibiotics and what is it, the beverages, that is what we can say alcohol, right? And organic waste in water, right? So organic waste in water, we can say that is BOD, biological oxygen demand. More the organic matter, more the organic waste, higher the BOD. When waste reduces, BOD increases if the waste is more. If the waste level, you can say that the organic matter level decreases, so BOD decreases. So that is what you have to keep it in mind. So biogas, which is a methane rich fuel. So A3, only in this option we have, so we can select A3, B4, C1 and D2, right? So in this, a very important thing, right? So D is the answer. And so study the following depicting, they study the following depicting the formation of curd from milk. Identify the missing part A and B. So here is a A, and we have to, what is it, so milk is incubated with curd, right? So that is what generally growth of the lab, that is by using inoculum, right? And production of, when lab will be used, generally that is which acts on milk sugar, right, lactose, and mainly that lab which acts on lactose and helps in the formation of lactic acid. So lactic acid, one we can consider in both options we have, and coagulation and digestion of milk protein, and finally leads to formation of curd, that is where the vitamin B12 level increases. So answer is C for 38th question. C for 38th question. So which of the following statement is incorrect? So word antibiotic is misnomer. Anti is a Greek word that means against, bio means life. So together they means against life, right? So in the context of disease causing organisms, whereas with the reference of human being, they are pro life, not against. So that is correct statement. So whenever we are going to consider that, this statement is correct. The flocks or masses of bacteria interwoven, that is what forms the mesh like structure, woven fungal filaments which form mesh like structure. So that is also correct. Components of biogas are methane 50 to 70%, carbon dioxide 30 to 40%, and traces of hydrogen, nitrogen and hydrogen sulfide. Even this is also correct. So none of these are incorrect. So answer is D. None of these are incorrect. So these bacteria grow anaerobically on cellulosic material. These bacteria grow anaerobically on cellulosic material, produce large amount of methane along with the carbon dioxide and hydrogen, are collectively called as methanogens. Example of such bacteria. So where we can say that the bacteria which will be methanobacterium, that is correct, and methanobravibacter and methanococcus. So that is what streptococcus we mentioned, methanococcus. So all of these are the correct answer for this 40th question. So biogas generation in 3 stages. So that is where we can say that generally the stage 1, where you can say that the polymers will going to break down into enzymatic breakdown of complex organic compounds into simple soluble monomers. So that is where we can say, we can select, that is the 41 question number. That is where the breakdown of monomers, here the first step. So protein all these in this, what is it, the breakdown of monomers. So in this breakdown of monomers, where we can see that is complete proteins in which amino acids and fats, cellulose, we can say that the hemicellulose, glucose content will be produced. And soluble compounds or monomers, right? Monomers are converted into organic acids by fermentation, causing microbes. That is after fermentation, right? So that is which will going to take place, and organic acids will be produced. Stage 3, organic acids are acted upon methanogenic bacteria. So on this, what is it, the methanogenic bacteria which will going to act, and those methanogenic bacteria which acts on organic acids and helps in the release of biogas. So that is all of these are correct, what is it, the stages in the biogas production, right? And read the following statement and select the correct option. So BOD represents the amount of dissolved oxygen that would be consumed if all organic compounds into the simple soluble were oxidized by microorganism. It represents the amount of dissolved oxygen, right, required to consume, that would be required to consume all those organic waste which would be present in the water. So that is where we can say that the generally the statement 1 is the correct statement, right? Statement 1 is correct, and I value of BOD indicates that water is highly polluted by organic matter. Statement 2 is also correct, but statement 2 is not a correct explanation for statement 1. So answer for that is B. So answer for this 42nd question is B. Both the statements are correct, but statement 2 is not correct explanation for statement 1. Right. Refer the given flowchart of sewage treatment and accordingly match column 1 and 2. So this is the what I have given there already, that is sewage treatment plan, right? So from this we can select the correct option. So 43rd question, right? The stage in which the physical treatment of sewage is done. So that is generally where we can say that the stage in which the physical treatment of sewage is done, that is where the primary effluent formation will be takes place, but in the primary treatment. So A we can write, that is 7th option. So A, that is 7th option, correct, right? So that is sewage physical treatment is done in, what is that, primary treatment. That is what we can say. Physical treatment also they can give physical treatment, and stage in which biological treatment of sewage is done. So that is generally 6th one. Here it is 7th and this is 6th, that is secondary treatment. And name of this sediment in primary treatment, sediment during primary treatment, that is I have already mentioned that is primary sludge. So that is 5. And then C5, it is carried in aeration tanks, that effluent, primary effluent from primary, what is it, the settling tank. So that is primary effluent. So that is D4, right? And name of the sediment in secondary treatment, so that is E, sediment which is called as activated sludge. So E2, right? E2. And next, site of flux growth, that is in the aeration tank. That is F3. Third option. And function of sludge digester, that is anaerobic digestion of activated sludge and production of biogas by anaerobic bacteria. that is methanogenic bacteria. So this is G1. G1. So A7 both we have, right? So these are wrong. B6, C should be 5. So in this we can get, whereas here in this is wrong. So all these are both second, third, fourth we can eliminate, and first one is the option, correct option for this the question. Enzyme which has a fibrinolytic effect. So I've already mentioned that the one which is obtained, right, where you can say that this streptokinase, which is using mainly the source which is generally obtained by streptococcus, that is streptokinase, that is actually TPA. What is the meaning of TPA? Tissue plasminogen activator. So once this tissue plasminogen activated plasminogen, right, so which generally which shows fibrinolytic effect. So fibrinolytic effect. So we know that generally fibrins which will be formed, fibrinogen converted into fibrin, so that leads to form a mesh like structure, and that is which helps in the formation of blood clot or coagulation. So that is, if blood clot takes place within the blood vessel, they're going to use this, generally the streptokinase, to bust that clot, a clearing blood clot because of tissue plasminogen activator, which activates and which as a fibrinol streptokinase which activates the TPA, and that activated TPA which is which shows fibrinolytic effect, which shows fibrinolytic effect. Which shows fibrinolytic effect. So in this fibrinolytic effect, they're going to dissolve, or I can say clear the the blood clot, and mainly to treat in the person who are suffering from myocardial infarction. So answer for this is 44th question, streptokinase. And the 45th question. So drug used for a patient A is obtained from organism B. Identify A and B above the statement. Select the correct answer. Drug A is used for patient A. Swine flu, Monococcus purpureus, that is wrong. AIDS, no, that is generally HIV. Right? Heart, penicillium, chrysogenum, that is wrong. So organ transplant, that is immunosuppressive agent, which is obtained from so patient A, a drug used for patient A, that is for organ transplant, which are already transplanted, and the drug is obtained by the Trichoderma Polysporum. So that is correct. So this drug Trichoderma Polysporum, I've already mentioned that, which is obtained from generally this fungi, which is a immunosuppressive agent. Immunosuppressive agent, that is cyclosporin A. Cyclosporin A, which is obtained to treat person who has got the organ from the other donor, the patient. Right. So next question, cyanobacteria are. So I already mentioned that these cyanobacteria, which have a capacity to show that is a photosynthesis, hence we can say that is phototrophs. They are autotrophs. Right. And then 47th question, cyanobacteria are autotrophic microbes. Right. So here you select the incorrect statement. In the 47th question, right, so select the incorrect statement. Which of the following statements are incorrect? We have to select. So cyanobacteria are autotrophic microbes, that is correct, widely distributed in aquatic and terrestrial habitats, correct. Anabina, Nastak, Acilitoria, photosynthetic nitrogen fixing cyanobacteria, that is also correct. The Tolipothrix, that is also blue-green, example for blue-green, which increase the rice production about 20%. That is what the Azula pinnata. Blue-green algae add organic matter to the soil and increase the fertility. That is also which acts as biofertilizers. And then what is it? The country, the biofertilizers are not available commercially in the market for farmers. So generally they are available. That is the incorrect statement. So only fifth one is the incorrect statement. So chemical substances produced by some microbes which can kill or retard the growth of other microbes are called as. So this is the last question we can discuss. That is the 48th question, where this chemical substance produced by some microbe which can kill or retard the growth. That is, what is that? Antibiotics. So best example, penicillin. So antibiotics which can be used. So antibiotics is a substance that will be used to treat the generally that is the infections caused by bacterium, right? So with this we can conclude this chapter. Thank you.